Hi all. Today we are going to start with the vitamin class. So what is a vitamin? Vitamin is a group of organic compounds and the recommend is very small. It performs a wide variety of biochemical functions. As it cannot be synthesized by the body, it has to be necessarily supplied in the diet. So it is a group of organic compounds required in a smaller quantity and it is not synthesized by the body and hence it has to be provided in the diet. Previously, it was thought that the vitamins, they have the amine group. Now, later researchers have proven that not all the vitamins have the amine groups and the letter E was dropped out and hence it is called as the vitamin. So how will you classify a vitamin? It is classified as a fat soluble vitamin and water soluble vitamin. So fat soluble vitamin includes vitamin A, D, E, K, and water soluble vitamins. It is classified as the B complex vitamin and non B complex vitamin, which includes the vitamin C. So it is classified as the fat soluble vitamin and water soluble vitamin. Fat soluble vitamin includes ADEK and water soluble it is classified further as B complex and non B complex vitamin, and under non B complex comes the vitamin C. So the B complex vitamins are further classified as the vitamins that releases the energy and the vitamins that possesses the hematopoietic activity and the other categories. The vitamins that releases the energy are thiamine, riboflavin, niacin, biotin, and pantothenic acid. And the vitamins needed for the formation of hematopoietic elements are folic acid and vitamin B12. Under the other category includes the B6, vitamin B6, it is pyridoxine. The active forms are pyridoxal and pyridoxamine. So coming to the difference between the fat soluble vitamin and the water soluble vitamin, based on the solubility, we know that fat soluble vitamins, they are soluble in fat, while water soluble vitamins, they are soluble in the water. So coming to the structure, the fat soluble vitamins, they have mostly isoprenoid units. What is an isoprenoid unit? It is an organic compound having the two or more units of hydrocarbon units, and each unit has 5 carbon atoms. They are arranged in a specific pattern. While the water soluble vitamins, the structure is heterogeneous. Coming to the absorption, the fat soluble vitamins need fat for its absorption, and bile salts. They enter into the lymphatic circulation, then reaches the blood. While in case of water soluble vitamin, they directly enter into the blood. Transport difference, the fat soluble vitamin requires the carrier protein to get transported in the circulation, while in case of water soluble vitamin, there is no transport protein needed. Exception here is vitamin B12. It needs a carrier protein for its transport in the circulation. Coming to the storage, this fat soluble vitamin, they get adequately stored in the liver and the adipocytes. So water soluble vitamin, as it gets excreted, they are not stored in a larger amount, and hence the continuous supply is needed. Except in case of vitamin B12, the storage form occurs. The functions of fat soluble vitamin are varied and they do not act as a coenzyme, except in case of vitamin K, they act as a coenzyme form. While in case of water soluble vitamin, most have the coenzyme activity. Excretion, the fat soluble vitamins, since they are mostly stored and gets let's excreted in the feces. Water soluble vitamins, they get excreted in the urine and hence it is considered to be a less toxic form. So toxicity of the fat soluble vitamin, when it is taken in the excess amount, hypervitaminosis occurs. In case of water soluble vitamin, since it gets excreted in the urine and it is not stored, the toxicity is less likely. Coming to the treatment, the single large dose prevents the deficiency, while in case of water soluble vitamin, and hence it is not stored and gets excreted, and regular dietary supply is needed for the water soluble vitamins. So these are the differences between the fat soluble and the water soluble vitamins. So we will see about the vitamin A. So objectives of learning here is you need to know on the dietary sources, various forms of vitamin A and their function, and what are the precursor form of vitamin A, the biochemical functions of vitamin A and its role in Wall's visual cycle, and what is the required dietary allowances, what are the deficiency manifestation, the causes for it, and its treatment. So coming to the vitamin A class, vitamin A includes the several related bioactive molecules. Actually, it is a collective term which includes the related bioactive molecules. If you see the structure, it is composed of the cyclohexane ring, right? So 6 carbon units, a cyclical ring structure is present, and a linear unit, which is the isoprenoid side chains. So it is composed of the beta ionin ring. The ring is called as the beta ionin ring with the isoprenoid side chains. As I have told earlier, the isoprenoid side chain composed of it is an organic compound having the 2 or more units of the hydrocarbon chain, and each unit composed of 5 carbons. They are arranged in a specific pattern. So there are two compounds, two groups having the vitamin A activity. One is the retinoid form. The second one is the carotenoid form. The retinoids are the preformed vitamins. They are derived from the animal foods of the animal origin. While the carotenoids, it occurs as a provitamin form of vitamin A. They are found in the vegetarian foods. So the two groups of compounds with the vitamin A activity are retinoids and carotenoids. Retinoids, they are derived from the animal origin, and carotenoids from the vegetarian origin. So we'll see one by one. The first about the retinoids. It includes a family of molecules. The includes both the natural and the synthetic derivatives with or without vitamin A activity. There are 3 forms of retinoids. One is a retinol, retinal, and retinoic acid. As you know, if the name ends with the OL, it has the alcohol group. If it has the AL group, it has an aldehyde group. Retinoic acid, it has a COOH group. So the retinoids, they are found in the nature from the food from the animal origin. They occur as the preformed vitamin A. So we'll see about the retinol. Retinol is a principal form of the vitamin A, and it has its role in reproduction. So retinol, it's stored in the animal tissues, especially the liver tissue is rich in vitamin A1. In the saltwater fish, it is present as the retinyl ester. What is retinyl ester? The retinol groups combines with the long chain fatty acid, usually palmitic acid, to form the retinyl ester, which is considered to be a very stable form of vitamin A. And the other form of retinol is dihydroretinol. This is otherwise known as vitamin A2. The difference between these two structures, retinol and dihydroretinol, is there is an extra double bond in the beta anion ring, right? So the retinol and dihydroretinol, since it has an extra double bond, they are susceptible to oxidation. Also, the isoprenoid units has 4 alternate double bonds, and it is present in the different isomers. Next, the next compound is a retinal. How retinal is formed is retinol when it gets oxidized with a reversible oxidation to form retinal. So this retinal, it has almost all the biological activity of retinol. So the biologically important component of retinal is, so the biologically important form of retinal is 11-cis retinal. So we know that the isoprenoid is a linear structure. There are 4 double bonds which are arranged alternatively, and because of the presence of the double bonds, there are many such isomers are possible, of which the biologically important form is 11-cis retinal. So the cis isomers are formed between the carbon atoms 11th and 12th position. This is considered to be the biologically important compound of retinal. And this retinal is essential for the normal vision. As we know, retinol is needed for the reproduction and retinal is essential for the normal vision. So this retinal is present in the plant sources. So it is present as a provitamin. So it has to get activated in the intestine. So there are so many carotenoids are present in the plant sources, of which the beta carotene is considered to be the most important form. It is a provitamin form of vitamin A. In the intestine, it is acted upon by the intestinal dioxygenase enzyme to form the aldehyde form of the retinoids, which is the retinal. In the animal source, it is present as the retinol, or it is present as the ester form, retinol ester. While in case of plant sources, it is present as a provitamin form. In the intestine, it has to get activated to form the retinal by the intestinal dioxygenase enzyme. So coming to the third form of the retinoids, which is the retinoic acid. How it is formed? The retinal, when it gets oxidized, it forms the acid, right? So it is a retinoic acid. It has some biological activity. While retinal has almost all the biological activity of retinol, while retinoic acid has only some biological activity. So this retinoic acid is essential for the cell growth and differentiation. Retinol is needed for the normal reproduction, while retinal is essential for vision, and retinoic acid is essential for cell growth and differentiation. So how the vitamin A molecules or retinoids, they get interconverted. So this retinol is present mainly found in the animal sources. So they get oxidized to form the retinal, which is an aldehyde form. When alcohol is oxidized, you know that there is a formation of aldehyde. So alcohol by dehydrogenases, alcohol dehydrogenases to form retinal. Also, the retinal is interconverted to retinol form by the intestinal reductases. So, reductase enzyme converts retinal to retinol, while retinol is acted upon by dehydrogenase enzyme to form the aldehyde form retinal. So it gets interconverted, while the retinal is oxidized to retinoic acid, but the reaction is irreversible. Till now, we have seen about the retinoids. Next, we'll see about the carotenoids. So there are two group of compounds which possesses the vitamin A activity, right? One is the retinoids. The second one is the carotenoids. These carotenoids, they are found in the plants. So it is composed of carotenes and its related components. So they are present as a provitamin form. It is considered to be a precursor form of vitamin A. It has to get activated in the intestine. On cleavage, it releases the aldehyde, retinaldehyde. Then it is interconverted to retinol and retinol to retinoic acid. So there are nearly 600 compounds are found in nature, of which only 50 has the provitamin A activity. The important groups of provitamin A carotenoids are alpha, beta, and gamma carotenes and cryptoxanthin, of which beta carotene is considered to be a main important plant source of vitamin A. So this is a structure of beta carotene. It has two units of vitamin A, right? So vitamin has beta ion ring and isoprenoid side chain. You could able to see the two units of vitamin A. On cleavage, it has to release the 2 units of vitamin A theoretically, right? So, but in the intestine, the conversion is inefficient and it releases a single molecule of vitamin A. It is acted upon by the intestinal dioxygenase enzyme to release a single unit of vitamin A. So if it is present in the beta carotene form, the vitamin A activity has only 1/12 that of the retinol. So we'll see about the absorption, storage, and its action on the uptake by the tissues. So in the diet, it is present either as a esterified form, retinyl esters, or as a beta carotene from the plant source. So this ester has to be hydrolyzed by the pancreatic enzyme hydrolases to releases the free fatty acid and then retinol. Retinol is absorbed by the intestinal mucosal cells. This beta carotene is absorbed by the intestinal mucosal cells as a provitamin form, where it is acted upon by the intestinal dioxygenase enzyme to release a single molecule of retinol, the aldehyde form of retinol. So they get reduced by the intestinal reductases to form alcohol, retinol. So this retinol combines with the palmitic acid or a long chain fatty acid and gets stored as a retinyl esters. Then it is incorporated into the chylomicrons where it is circulated into the lymphatic circulation, then reaches the liver. So in the liver, it is stored as the retinyl palmitate. The storage form is the retinyl ester. So liver holds about 90% of vitamin A reserves. And whenever there is a need, this retinol palmitate is hydrolyzed to release the retinol. So this retinol is transported in the circulation by combining with the retinol binding protein. Also, it combines with one more molecule called as transthyretin, which is a pre-albumin molecule. So it forms a larger trimolecular lipoglycoprotein complexes. So and hence this prevents the loss through the glomerular filtration. So this retinol binding protein along with the retinol, along with the transthyretin molecule, it is recognized by the cellular surface receptors. So the cellular tissues, they recognize the retinol binding protein, retinol, and the transthyretin. So only the retinol molecule enters into the cell. So in the cell, it has one more cellular retinol binding protein where it reaches the nuclear receptor. So this retinoic acid is recognized by the nuclear receptors. There are two nuclear receptors. One is the retinoic acid receptors, RAR, and retinoic acid X receptors are there, RAR and RXR receptors. So this retinoic acid in the cell is recognized by the two nuclear receptors, which in turn binds to the response elements of the gene, which causes the activation of the gene, resulting in transcription of the mRNA, ultimately translation of the necessary proteins needed for the cell growth and differentiation. So the efficiency of absorption when it is present in a preformed form, as in case of retinol, it is 90% absorption. When it is present in the carotenoid form, the absorption efficiency is only 9 to 22%. So coming to the biochemical function of vitamin A, it takes parts in visual cycle. So vitamin A is a component of photoreceptor complexes of the retina. In the retina, there are two types of photoreceptor cells are present. One is a rod, then the second type of cell is the cones. Rods and cones are present in the retina, and the visual pigment that is present in the rod is a rhodopsin, and the pigments present in the cones are one for blue, which is the cyanopsin. For the green color, it is iodopsin, and for the red color, it is called as porphyropsin. So there are three photo visual pigments are present in cones. One is the cyanopsin, iodopsin, and porphyropsin, and the visual pigment in the rod is the rhodopsin, which plays a pivotal role in visual cycle. So when a light reaches the retina, hits the retina, before it reaches, it has to travel through the cornea, aqueous humor, pupil, and vitreous humor. So in the retina, as you know, you have two types of cells. One is the rods and cones. Rod is responsible for the visual cycle, and cones are for the colors. So the pigment are rhodopsin, cyanopsin, iodopsin, and porphyropsin. So the visual pigment is composed of two components. It is a conjugated protein composed of retinal, which is present in the 11-cis retinal form, and the opsin, which is a protein. So it is composed of opsin and retinal component. Retinal is present as the 11-cis retinal, and opsin is a protein. So each of the four pigments, they observe a certain wavelength, and rhodopsin, they observe the middle part of the spectrum that comprises 500 nanometer. So the rhodopsin is composed of the retinal component and the opsin, right? So it is a conjugated protein, and retinal is in the 11 cis form. It forms a Schiff's base with the epsilon amino group of the lysine present in the opsin. So it is a conjugated protein composed of retinal and opsin, and the retinal is, they form a Schiff's base with the epsilon amino group of the opsin. So next we will see about the Wald's visual cycle. When a photon, that is a light, hits the retina, so in the retina you have the visual pigment in the rods, that is a rhodopsin. They get activated and get which causes a release of the opsin and levensis retinal. This levensis retinal is isomerized to form the all trans retinal. So levensis is isomerized to all trans retinal. So immediately there are many intermediates are formed. One is a beta rhodopsin, lumirhodopsin, meta-rhodopsin 1 and meta-rhodopsin 2. So this, during the conversion, the opsin, there is a structural reorganization of the opsin, which in turn causes the changes in the resting membrane potential, which in turn generates the nerve impulses. So signal transduction occurs. Okay, so some amount of all trans retinal in the retina is converted to levensis retinal, which combines with the opsin, and hence it is regenerated. While majority of the all trans retinal, they travel through the blood, reaches the liver. In the liver, it gets acted upon by the dehydrogenases enzyme to get reduced to trans retinol. So this retinol get isomerized by the retinol isomerases in the liver to form cis retinal. So trans retinol is isomerized to cis retinol. This retinol is oxidized by the dehydrogenases to form cis retinal. This retinal reaches the retinal epithelium, combines with the opsin to form rhodopsin, and hence it is regenerated. So this is a picture showing the rod cell with the photopigment rhodopsin. When a photon hits the rhodopsin, it causes a structural reorganization. This rhodopsin is associated with a one more protein called as transducin molecule, which is a G protein complex. So this G protein is composed of alpha, beta, and gamma protons and gamma subunits. And this G protein in the inactive state, it is GDP bound. When it gets activated, what happens? This GDP is converted to GTP, and this GTP alpha subunit is dissociated from the transducin complexes. This alpha subunit of the transducin with the GTP, they activate the phosphodiesterase enzyme present in the cell membrane. This in turn causes the conversion of cyclic GMP to GMP. So this catalyzes the conversion of cyclic GMP to GMP. So this, the decrease in the cyclic GMP causes the CGMP gated sodium channels to close, and hence the sodium level inside the cell falls, resulting in hyperpolarization. This results in neuronal signaling. Thus the visual impulse. One more picture explaining the sequence of events in visual cycle is when a light hits the retina. There is a photopigment called as rhodopsin in the rod cells, right? So this rhodopsin is composed of the levensis retinal and the opsin protein. It is a conjugate protein. When a light hits on it, there is photoisomerization of the levensis retinal to all trans retinal. So this, during its conversion, there are several intermediates are produced. One is the meta-rhodopsin 2. This in turn causes the structural reorganization of the rhodopsin molecule. This rhodopsin is associated with the one more molecule called as transducin. The structural changes is transferred to the transducin molecule. Normally, in the inactive state, it bounds with the GTP complex. When it gets activated, the GTP bound alpha subunit of the transduction dissociates from it and gets active. They activate the phosphodiesterase enzyme, which in turn catalyzes the conversion of cyclic GMP to GMP. So the cyclic GMP gated sodium and calcium channels, normally it causes the influx of the sodium channels. When the level of cyclic GMP is decreased, this in turn causes the closing of the sodium channels and hence causes hyperpolarization of the membrane, which in turn results in the neuronal signaling. So our visual system is responsible for responding to the color and light within a few milliseconds. How it is possible? This is due to the fine tuning mechanism that is present. The one such theory is the transducer molecule has the intrinsic GTPase activity. So this GTPase catalyzes a conversion of GTP to GDP. When it is bound to GDP, it becomes inactive, and hence it decreases the activity of the phosphodiesterase molecule, and hence there is elevation of the cyclic GMP. This in turn causes the opening of the sodium channels. The other mechanism is, so when the metorhodopsin is phosphorylated, there is one more molecule called as arrestin. This arrestin will go and bind with the phosphorylated rhodopsin molecule, metorhodopsin molecule, and hence it stops the signaling effect, neuronal signaling. The third mechanism is there is activation of the guanyl cyclase enzyme, which catalyzes a conversion of GTP to GMP, and the level of cyclic GMP in the cell is increased, which in turn causes the opening of the sodium channels, resulting in the stoppage of the signaling. So next we'll see about the dark adaptation time. For example, when you walk in a bright sunlight and go into the dark room, as in case of the cinema theater, right? So what happens, initial few seconds, you couldn't able to visualize any object in front of you, right? So this is due to the rhodopsin molecule has to get resynthesized, and it takes a few milliseconds. So the resynthesis of rhodopsin occurs only few milliseconds after it gets activated. And in case of vitamin A deficiency, what happens? The dark adaptation time is increased. Till now, we have seen about the biochemical function in the visual cycle of vitamin A. So let's see about the other biochemical functions of vitamin A. So they are needed for the normal reproduction, and retinol is in the retinol form. They are essential for the normal reproduction. These retinol and retinoic acid acts like a steroid hormone, and retinoic acid is responsible for cell growth and differentiation. As you know, in the blood, it is transported as a retinol form, binds with the retinol binding protein, combines with the one more molecule of transthyretin, which is a prealbumin molecule. Whenever there is a need, whenever our body needs the cellular growth and differentiation, this retinol is taken up by the peripheral tissues, and this in the cellular tissue, it gets bind with the cellular binding retinol binding protein. It gets oxidized to form the retinal component. So all trans retinol is taken inside the cell. It gets oxidized to form all trans retinal, which in turn further oxidized to form the retinoic acid. This retinoic acid is recognized by the nuclear receptor. So the two nuclear receptors are retinoic acid receptor and retinoid X receptors are there. Retinoic acid receptor, they bind with the trans-retinoic acid, while in case of the retinoic acid X receptor, they bind with the 9-cis retinoic acid. They form the dimer. They in turn go and bind with the hormone response elements, or the response elements in the gene, which in turn causes the transcription of the gene, resulting in the protein need synthesis of the protein needed for cell growth and differentiation. So the other function is they maintain the normal epithelial tissue. They maintain the integrity of the normal epithelial tissue. In case of vitamin A deficiency, what happens? There is drying of the epithelium, resulting in the prone to infections. The other function is the retinyl phosphate is needed for the synthesis of the glycoproteins, which is a component of the mucous membrane. So in case of vitamin A deficiency, there is drying of the mucous membrane of the respiratory tract, gastrointestinal, as well as the urogenital tract. Also, vitamin A is essential for mucopolysaccharide synthesis in the extracellular matrix. So also vitamin A is needed for the maintenance of the immune system. In case of vitamin A deficiency, what happens? There is a keratinization of the mucous membrane, which leads to susceptibility to frequent infections. This vitamin A is also needed for the synthesis of the transferrin molecule. This transferrin is an iron transport molecule. In case of iron deficiency anemia, vitamin occurs, that is vitamin E deficiency results in iron deficiency anemia because of the decreased in the synthesis of the transferrin molecule. Also, it acts as an antioxidant molecule, also an anti-cancer drug, anti-cancer agent. So how they act as the anti-cancer agent is by blocking the tumor promotion, inhibit the proliferation of the tumor cells, inducing the apoptosis of the cell. That is, it causes the shedding of the tumor cells, and it also has a natural antioxidant property. This vitamin A, they remove the free radicals. Also, beta carotene reduces the risk of myocardial infarction. As you know, rods are responsible for the dim vision, and the cones are for the color vision. Till now, we have seen about the biochemical functions. Next, we see about the deficiency manifestations of vitamin A. First, mostly it involves the eye. The first change is a night blindness, otherwise known as nyctalopia. This is the earliest symptom of vitamin A deficiency, in which the patient couldn't be able to see the object in the dim light. So this is the first change of vitamin A deficiency. So there is impairment in the dark adaptation. The first change is a nyctalopia. Also, the Bitot spots occur. So you could be able to see the triangular white plaques, right? So they are present on either side of the conjunctiva, and it is usually bilateral. So there is a gray white triangular plaques on either side of the cornea. Usually it is bilateral. The next change is the xerophthalmia. When the night blindness is left untreated, it leads to a change called as xerophthalmia, in which the cornea and conjunctiva is dry, thickened, wrinkled, dull, and glazy, right? So it loses its normal transparency. This changes is known as xerophthalmia, so which leads to blindness. Next change is the keratomalacia, which means softening of the cornea. There is the ulceration and necrosis of the corneal epithelium. Ulceration is the breach in the normal epithelium. If the xerophthalmia persists for a longer time, it leads to a condition called as the keratomalacia, in which there is ulceration and necrosis of the corneal epithelium. The degeneration of the corneal epithelium leads to later vascularization, which leads to the opacity of the cornea. So this keratomalacia is the most important cause of preventable blindness in developing countries. It usually associated with the protein energy malnutrition, and hence as a prophylactic measure, children under the age of 1 year, they are provided with a single large dose of vitamin A. So the last stage is a perforation of the cornea, which leads to total blindness. The stages are eye changes are the first is the night blindness, second is the xerophthalmia, the next is the keratomalacia, next is the total perforation of the cornea. These are the eye changes. Next, we will see about the other deficiency manifestations. The changes that occurs in the skin is a follicular hyperkeratosis, otherwise known as phrenoderma. Because of the mucosal epithelium integrity is lost in case of vitamin A deficiency, it becomes thickened and keratinized. This it has to be confused with, this is usually confused with the acne. So these are the follicular hyperkeratotic changes of the skin in case of vitamin A deficiency. Here there is hyperkeratinization of the epithelium. Also, the patient presents with a delayed growth. This is because there is decreased synthesis of the chondroitin sulfate. Also, there is decreased synthesis of the protein, glycoprotein, as well as the reduced immunity. Next, we will see about the causes for the deficiency. It is classified as a primary cause and a secondary cause. The primary cause is due to decreased intake of vitamin A. while the secondary causes are so many. It is due to the obstructive jaundice. In obstructive jaundice, there is defective absorption of lipids. Usually the fat soluble vitamins, including the vitamin A, they get absorbed along with the vitamin A and bile salts, right? So in case of obstructive jaundice, there is defective absorption of the lipids. In case of cirrhosis, cirrhosis is a condition in which the causes are varied, in which there is a fibrous scarring of the liver architecture, and the retinol binding protein is synthesized in the liver, and this retinol binding protein is needed for the transport in the circulation, right? So if it is not synthesized, it is not circulated. So there is impaired retinol binding protein production in case of cirrhosis, and this is one of the secondary cause for the vitamin A deficiency. The third cause is a chronic nephrosis, in which the retinol binding protein, they get excreted in the urine. So in vitamin A deficiency, there is reduced retinol binding protein. The secondary cause is chronic nephrosis, in which retinol binding protein is excreted in the urine. In case of chronic alcoholism also, there is vitamin A deficiency. This is due to hepatic injury. Also, it competes with the dehydrogenase enzyme. Alcohol competes with the dehydrogenase enzyme, leading to vitamin A deficiency. How are you going to assess a vitamin A deficiency? You can do the dark adaptation test. In case of vitamin A deficiency, it is going to get elevated or increased. Next, you can assess the retinol binding protein level. In case of vitamin A deficiency, the RBP level is going to get decreased. And you can assess the serum vitamin A level. Normal level is 25 to 50 microgram per deciliter. And the method of estimation is by the Carr and Price reaction, in which the vitamin A is extracted with the chloroform, and it gets bind with the antimony trichloride to form a blue color complexes. The final color complex is unstable, and hence it has to be immediately measured spectrophotometrically at 325 nanometer. Next, we'll see about the daily recommends of vitamin A. It is expressed as the retinol equivalence. So recommended daily allowances is expressed for vitamin A as retinol equivalence. One retinol equivalent is equal to 1 microgram of retinol. So previously it was expressed as international units, now as a retinol equivalence. For men, it is needed as 1000 retinol equivalents. For women, it is 800 retinol equivalents. So the higher recommends are needed for growing children, pregnancy, and in case of lactation. So one retinol equivalent or one retinol activity equivalent is equal to 1 microgram of retinol. When you want to express in terms of carotene, beta carotene, one retinol equivalent is equal to 6 microgram of beta carotene, and one retinol activity equivalent is equal to 2 microgram of beta carotene. Next, we'll see about the dietary sources of vitamin A. So it is present as a retinol form or a preformed vitamin A in case of the animal sources, right? So it is present richly in the liver, milk, butter, egg, and fish. In the green leafy vegetables, yellow to orange pigment fruits, they contain carotenoids. So it is a provitamin form. In the intestine, it has to get activated. And it is also rich in green leafy vegetables like spinach and amaranth. Hypervitaminosis occurs when a patient consumes the vitamin supplements without any proper medical advice. He is going to suffer from the hypervitaminosis A. This hypervitaminosis A commonly occurs in the people living in the Arctic region who usually consumes the polar bear liver, which contains a rich quantity of vitamin A. So in case of hypervitaminosis A, the patient suffers from the symptoms of increased intracranial tension, which are the headache, nausea, vomiting, diarrhea, the drowsiness, vomiting, anorexia, and irritability. Also, liver enlargement occurs in case of the children, and exocytosis of the long bone occur. This is the bone spur formation in the long bones. There is extra growth formation in the surface of the long bones. Also, the patient suffers from excessive dryness, desquamation, and alopecia. So there is one more condition due to increased intake of beta carotene called as the hypercarotenemia, in which the patient's skin is stained with the orange color, right? So this has to be differentiated from the jaundice. In jaundice, there is elevated serum bilirubin level. The difference between these two conditions is the you will need to look in the sclera. So in the hypercarotenemia, the sclera is normal. There is no yellow staining, right? So in case of jaundice, there is the yellow staining of the sclera also. These are my references. Thank you.